Hi everyone, welcome to lesson 1.2. This is scientific methodology and context. So as we go through this lesson, remember to fill out your guided notes. Again, you can either print those and fill them out if you like. You can type directly into them. If you want to write your notes in a notebook, you can also do that as well. But you want to make sure that you are filling out your notes as we go through this lesson because you will want those notes for both your vocabulary warm-up quizzes, your blend quizzes, and other various things as we go through the year. So as you listen, make sure to fill in your notes. So today we're going to be talking about the scientific method as well as kind of looking at designing experiments. So the scientific method you have probably seen before. Here's kind of a nice graphic. And here is the actual scientific method itself. So we start with an observation, or the act of noticing and describing events in an orderly way. We then make inferences, which are logical interpretations based on what scientists already know. A hypothesis is a tentative scientific explanation that can be tested. Notice that is underlined, that is important. Hypotheses have to be able to be tested by further observation or experimentation. Now remember, whenever we go through a presentation and there is a term in blue, like hypothesis, it means that that is a vocabulary term. If you go back to Blend, remember you can find your vocabulary list, and those are the terms that will appear on your vocabulary warm-up. So anytime you see something in blue, this is the definition you want to make sure that you have down for those vocabulary warm-ups or quizzes. Testing hypotheses often involves designing experiments that measure factors that can change, or also known as variables. The scientific method is the general style of investigation that we use to conduct research, just because it is a good way to go step by step. And then during the course of an investigation, hypotheses may have to be revised, experiments may have to be redone several times before drawing conclusions. So in designing a controlled experiment, kind of pause here and think to yourself. In an experiment where you're testing a hypothesis, how many variables should be changed, and why is it important to control variables? So pause here, think for a moment or two. So here are variables. The independent variable, again, both of those independent and dependent variable terms are in blue. Make sure that you write down that term for your vocabulary list. So the independent variable is the variable that you deliberately change in an experiment. This is what you are testing. This is also sometimes referred to as the manipulated variable. The dependent variable is observed and changes in response to the independent variable. So this is often the results, sometimes referred to as the responding variable. Oftentimes in an experiment you will also have a control group, and that is a group that is exposed to the same conditions as the experimental group, except it is not given the change that the independent variable was. Now, to talk about data, right? So you go through your experimental observations and then you're going to be gathering information called data. There are two main types of data: quantitative, which is where numbers are obtained by counting or measuring. And here are a few examples of quantitative data, right? Height, weight, number, volume, things that you kind of can measure and have an actual number attributed to it. Qualitative data is the type of data that is descriptive and involves characteristics that usually cannot be counted. So some examples: texture, taste, smell, color changes, right? Now in an experiment, we will almost never ask you to taste or smell something, but it is important to note that to add to your qualitative data. Now, here's our first example. Okay, the hypothesis is bean plants will not sprout if they are not given water. Okay, so you're going to go ahead, pause here, just kind of examine this diagram and kind of think to yourself, what is this experiment, what is this diagram showing? Okay, so now here's an example, a check for understanding. You can again pause here, and I want you to try to answer these questions by yourself. All right, now there's a lot of text on this slide, but we're going to kind of talk this one out. So what variables were kept the same in order to not affect the experiment? Okay, now not all of these were shown on that one picture, but here are a few things that we can assume. So they kept the number of bean seeds per pot the same type of pot, the same soil, same light exposure, same location. Okay, What would be some examples of quantitative data collected? The amount of water dispensed, amount of light exposure, the number of bean seeds, number, temperature, length of time, right? Amount. So remember, quantitative data is the actual number of something. What would be some examples of qualitative data? Okay, so observations about the soil, was it wet or dry? Observation about the bean seeds, such as color. Observations about the sprouts, location. So again, things not necessarily that have a number attributed to them, but are still important to note. What would be some additional experiments or tests that could be done to help support the original hypothesis, right? So if you set up additional pots and vary the amount of water given. So maybe have no water, light water, medium water. You could also set up additional pots and keep the amount of water the same but vary the light. Why is it important to have a control group? So the control group kind of acts as your baseline test to see what would happen if you did not expose the experiment to the independent variable. So in other words, for this experiment, what if it wasn't the water that was affecting the bean plant sprouting, right? So is my experiment being affected by my independent variable, or is something else affecting it? Would it be a good or bad idea to repeat the experiment multiple times or perform multiple trials? So we always, always, always try to reproduce or replicate our observations. Therefore, you can set up several control and experiment groups, and the more times you can repeat the experiment, the better. That will add to your data. Why is it important to collect a lot of data? Remember, experimental results will always vary. There's pretty much never going to be a time where you repeat an experiment and get the exact same results. So a larger sample size and large amount of data is always important for scientific methodology. It means that you can more reliably analyze variation within each group, and you can evaluate the differences between the experimental and control groups. Okay. Scientific theories and laws. So a theory applies to a tested, highly reliable scientific explanation of the natural world that unifies many repeated observations. Remember, a hypothesis is an explanation that can be tested, but it is not highly reliable. Therefore, it could be rejected with data collected. What's the difference between a scientific law and a scientific theory? Theories do not become laws. Ever, ever, ever. Theories do not become laws. Scientific laws are concise. They are specific descriptions of how some aspects of the natural world are expected to behave in a certain situation. Theories are much more dynamic and complex. So to kind of help us understand the really big difference is looking at what are a few examples of laws and theories. So for laws, ideal gas law in chemistry, conservation of energy, law of gravity. Theories, we have the theory of evolution and cell theory. Okay, so looking at those two, laws often have some type of like mathematical or quantitative component to them, whereas theories are more so supported by a lot of highly reliable scientific explanations. Okay, so theories tend to encompass a greater number of ideas and hypotheses than laws. They are usually fine-tuned throughout the process of science. Once a theory has been thoroughly tested and supported by many lines of evidence, it may become the dominant view among the majority of scientists. So as we know, the theory of evolution is supported by most scientists, okay? But it is never going to be called the law of evolution. And here's a great article down here if you'd like to click on that so you can read a little bit further. Okay, so now we are reaching the end of our first lesson. So here at the exit ticket, I want you to pause, think, and try to fill this out by yourself. All right, here is the exit ticket so you can check yourself. So remember, theories encompass a greater number of ideas and hypotheses. They're constantly fine-tuned, and scientific theories do not become laws. Okay, what's the difference between a hypothesis and an inference? This is number 2. So scientists make inferences, which is a logical interpretation based on what scientists already know. A hypothesis, again, can be tested, okay, by further observation or experimentation. Number 3, the dependent variable is the variable you are observing and changes in response to the independent variable, okay? Number 4, so there are many correct answers for this question. The answers I have up here, if yours is different, does not necessarily mean it is incorrect. Just kind of a basic hypothesis: if the amount of fertilizer is increased, the plant growth will increase. The independent, or the variable that you are manipulating, is the amount of fertilizer. The dependent, or the one that you're observing, is the plant growth. Okay, and the control group would be plants given no fertilizer or not given that independent variable. And number 5, why is it important to collect all data even if it doesn't fit your proposed hypothesis? So this question is really important. You never, ever, ever throw out data, okay? So you need to be careful to avoid errors in data collection and analysis. The larger the sample size, the more reliably you can analyze variation within each group. Even if data doesn't fit your hypothesis, you still have to record it and then repeat your experiment to find potential sources of error. You may also need to reevaluate your hypothesis if you have a large margin of error. So again, data, even if it doesn't fit your hypothesis, might be telling you something important. So now that we've done this first lesson together, you can go ahead, finish your guided notes, go back and make sure that you have all of your vocabulary recorded from this lesson. And thank you very much for listening.